So that was an emergency bio, because Bell found a bio at the University of Manchester, which was like 20 years out of date. Like, no. And we realized that Sam, the tech guy, was still, he was 4 years old when that bio was made. So so it was tailored for a slightly different project or talk. Okay, so this is the Youth Bioinformatics Symposium, although I see one or two people perhaps aren't 18 in the audience. And I was told that what you really wanted to hear about was what do I do in bioinformatics and how did I get there? So that you can basically see, how can I travel there too, right? And while I'm here, the reason I'm here is because I'm one of the co-chairs of the overall conference that you're all about to enjoy. So you're going to hear about my journey in computational biology and bioinformatics. And in particular, I'm going to talk to you about digital infrastructure. And what do I mean about that? So, okay, so bioinformatics, if you're here, this is what you think bioinformatics is about. It's about data, driven particularly by data, managing data, problem solving and doing data analysis, and then doing discovery and going to a loop like. So it's all about data. And what we're talking about when we're talking about data is complicated, different kinds of data from different resources, different modalities of data, different kinds of omics of data, proteomic data, transcriptomic data, phenotypical data, all different kinds of data from chemistry, data from public health, data from omics and genomics and from instruments and so on, all different kinds of data, all integrated. And the picture here I've got is interactomic. Every time there's a new piece of kind of method or we stick an omics on the end of it just for the sheer hell of it, right? So this is what we're really trying to do when we're managing our bioinformatics. Before you can do anything cool, you have to get the data. So there's two things I really wanted to emphasize then that are the bedrock of what I do in bioinformatics. The first is it's a data-driven science, right? So first you have to have a lot of data that we manage at many different levels. We manage it as public archives, as big repositories managed by big organizations like the European Bioinformatics Institute, for example, or managed by specialist organizations or countries at the national level, or managed within your own project in your own university or your own institute. But we're talking about data sets and registries and knowledge bases at these different levels. The key thing here is it's all distributed, it's all different, it's all siloed, it was never designed to be linked together, everybody stands up their own thing, and everything in it needs to be described and linked together. And people hate describing stuff, and then they want it linked together. So that's one of the first issues. And then the second is we need software. We need software in order to be able to do the analysis. So we build data preparation pipelines, we do data cleaning pipelines, we want to be able to analyze data from different data sets, we want to do simulations and AI predictions, and we need visualization tools. Like I've got a picture up here, and they need to run on cloud computing. They need to run on HPC. They need to run on distributed clusters in your own university. They need to run on your laptop, right? So we're talking about automation, and we're talking about, is it reproducible? And so can somebody else do it too? Now we're all with added AI. So there's going to be an AI bingo card for the whole conference. Every time you hear the word AI or LLM, you have to fill out a bingo card. The second thing that drives all of my kind of background and research is team science. So this is a team effort. You can't manage all the data yourself. You can't generate all the data yourself. You can't do the analysis on just your data. You have to link it to all the referenced data sets. So this is a collaboration effort. Bioinformatics is a collaboration effort. You can't just be like a historian sitting there doing your own individual research and then publish your book. It isn't the way it works. This is a team sport to do bioinformatics, but it's made up of individual efforts. So even when you have a big collaboration, a big project, you're still talking about your individual effort and your individual contributions. And it's a team of multiple skills. Biologists, sure. Bioinformaticians, that's all you guys. AI specialists, data scientists, data curators, managing those data sets, stewarding those data sets, research software engineers building the platforms for that. So it's a whole bunch of different people working together. And this is a picture from one of those biodiversity projects I was involved in. And this is a mixture of skills, right? And all of them need to be respected and work together. So in my career, I've worked in numerous different areas in biomolecular simulation, in systems and synthetic biology, across omics worlds, in industrial biotechnology with bioreactors, COVID surveillance, cancer, and so on. So, for example, one of the projects I was heavily involved, well, we were all involved in the COVID era, was a project, a big European project called BioCovid, which was about preparedness for infectious diseases. And this was mobilized during the period of COVID. We had all of the different data hubs, mostly across Europe, but not all entirely across Europe, which we had to connect all those different data sets. We had to do standardized way of describing the data. So some of our work was in how do you do that standardized description. Then we need to do analysis that was repeated analysis as we got new sequences, repeat the analysis, and then make that available. So all that analytical work on the automated monitoring, generating information for national dashboards, was all to do with analysis over all these different multiple databases, which we now previously described. And then things like producing toolkits, like giving guidance. So this is how do I know what I should be doing? Or disease maps, so that I can share models of the COVID interaction mechanisms. In all of these platforms, towards the end, these are all the ones I helped build. And that's what I'm going to be talking about a little bit. But I don't just do biology and life sciences, I also do biodiversity. So here are other things that I'm doing, things like platforms for being able to support ecosystem service modeling, or looking at the invasive alien species. So how do we deal with the invading species of certain, like crabs in area of the Baltic because of the changes in climate, or what do we do with crop modeling as climate changes, the wild crops and their existence in the current, the world as that changes, as climate changes, but also things like the ERGA project and the Biodiversity Genomics Europe project, which seeks to try to sequence species across Europe and to build barcodes of those species. So we're involved in those projects. And actually, a really slightly more off the track project is doing things like digitizing natural history museum collections. So here's a bee and some words written in handwriting, 19th century handwriting. How do we turn that into digitized material rather than the physical artifact so we can share it? Particularly as these decay, the glue decays, so we have to be able to recover. So I do all this kind of stuff. So this is what we might call knowledge turning at scale. So I've got a database and I want you to be able to use it. So how do I make it so that you can use it so we can turn the knowledge? How can we move knowledge between different organizations? How can we make it so that different computational platforms can access that? How do we make it so that everybody can access those computational platforms? So this is all about moving knowledge between systems and between people. Do you see what I mean? I'm now, because there's so few of you, it's like being in a class, except I don't know your names yet, but I will by the end. Okay, so, and really being able to move that information, a pan-organization, pan-country, everywhere all the time. So this is actually the principle of open science. So I'm one of the sort of drivers behind the world of open science. So that's open source code, open methods, so how did you do things, open data, publicly accessible research literature, the whole trying to make things as open as possible. What I won't be talking about is I'm also, which Bill didn't mention, the co-lead of Federated Analytics for Health Data Research UK, which is a big UK organisation that shares health data, analyses health data, and there it isn't open. There it's actually very controlled. So, but I'm not going to talk about that. I'm going to talk about other work. And in particular, I'm going to talk about something called Elixir. So Elixir is the research infrastructure for Europe, which is a pan-national government organization in order to be able to say, how do we manage the data sets of Europe, and how do we manage access to data sets if you're European? How do we do analysis? How do we make computational infrastructure available to people? How do we train people? How do we build a network of experts? So we have 1,000 experts in this network. It has 20, well, it depends on how you count it, but at the moment, let's call it 25 countries, including the European Bioinformatics Institute, which is a country in its own right in this. It has numerous communities associated with it, like rare diseases, proteomics, structural bioinformatics, metagenomics, and so on. They're all forms for all people to come together to how do I standardize my data? How do I analyze my data? How do I prepare and support my data sets? This is what this is all about. And I'm the head of the UK node, which has 28 organizations, soon to be 29 in it, where we do this at the national level and then feed that into Europe. So it's a big organization, lots of activity. And I've also established something called Biofair, which is a biocommons for the UK. So it's a national infrastructure for people who get grants in any kind of form of life sciences, a place they can go in order to be able to share data, learn how to make their data standardized, learn how to annotate it, and have national resources to analyze it. And that's a 34 million investment in its first incarnation. So that's another thing that I've led. And here's an example of this is a meeting. There's me with the arrow at my head. This is a gathering of the Elixir All Hands part of it. So it's a big organization. Now, why am I telling you all of this? Because I'm leading all these different things. So, here's my doctorate thing. Here's me getting prizes and on magazines, and I got a CBE from the Queen. Well, it was from the Queen, which means, 6,000 people die, I get to be Queen. That sort of thing. So I've got all this stuff. I have a biology high school A-level from 1979. That's the only biology I've got. And I'm the head of the UK Bioinformatics European node. And I'm co-running this conference. Go figure. Right? I'm not a biologist. I'm a computer scientist. That's what I do, right? And that's what I kind of wanted to say to you as a community is, well, I must get a degree in biology to do bioinformatics. Well, sometimes you don't have to, right? Because we have to have all different kinds of talent in the bioinformatics community, not just technically, but also the soft skills of bringing people together and solving problems. And that's something I would like you to take away from this, this talk. Okay, I also never intended to be an academic. So here's me in the 1980s when I was, cool. I had a teaching lectureship. I did a lot of stuff with women in computing. You'll discover there aren't enough women in computing. There's me in 1979. That's what it looked like when I went to university, right? This is what we worked on. I was actually taught by Tom Kilburn. Has anybody ever heard of Tom Kilburn? Tom Kilburn invented the first stored program computer in the world, right? And I was taught by him that. So you've touched the person who worked with Turing, right? Turing did the programming. Yeah, that person taught me. So, and I know I'm old, but not that bloody old, right? So it's really, it's a young discipline, right? Why did I become an academic? Well, that's a good question. I married somebody. Anyway, there we are. I married my professor. I'm very efficient. He'll do. Anyway, he won't be watching this video, I can say that. So what I want to point out here is that, look at this iceberg, right? At the top, there's all the cool bioinformatics that you'll hear about from everybody. And underneath, there's the data stewardship, there's the software development, there's the preparation and the data wrangling and the instrument pipelines and all of the rest of it, some of it using machine learning and new novel things and so on. But to do fancy, cool bioinformatics and biology, you need all of that underneath. And that's the bit I do. Right, that's the bit that we lead on. And, this is another picture riffing on the same sort of theme. This is from Julia Lane, who's a leader of AI thinking in the US. She has this fountain picture that says, you have this sparkly fountain of all this AI knowledge, that we'll hear later from our keynote speakers and others. But underneath, you need some plumbing. Otherwise, there isn't any fountain. And that plumbing is all about findable, accessible, interoperable, and reusable data, right? And the infrastructure, the computing infrastructure to support it, and the people skills to support it. So I would say that if I had to position myself, I would say I'm a translational research in computer science. I'm a researcher who's a translational computer scientist, because I'm actually a computer scientist. My degree is in computer science. I'm a professor of computer science. And I would do what I would class what I do as empirical research. So my empirical research is, the theory is in kind of computer science, the applications are computational science, and the practice is software engineering. So you can come up with great ideas of how to represent knowledge in theory, but then you have to make them work in practice, right? And that's the translational piece. Now, I'll give you a sort of an example of that. So if I say a lot of the stuff I'm talking about here is digital research infrastructure, so that's how do I store data, how do I do analysis? In order to be able to do bioinformatics, in order to be able to do biology, this is what I do. I'm a computer scientist, I'm a research software engineer, because computer science is the theory behind all the computing stuff. But the difference between a computer scientist and a software engineer is a computer scientist, it doesn't have to work. And a software engineer it does have to work, because somebody else is going to be able to use it. But at the top, I people wrangle. Do you remember that big picture of all those people? I know the names of 70% of those people. And I work with them on a daily basis to get things to work together, to combine data sets, to make portals, to build infrastructure. So that's what I do. And so this is a principles and theory to products and practice. So I've actually got my research software engineer t-shirt here, which you can see is a unicorn, because we're unicorns. Okay, so how did I get into bioinformatics then? So the next few slides, I'm going to talk about how I got into bioinformatics. So at the top, the pale blues are the sort of technical work I do as a computing person. I do knowledge management or knowledge and what's called metadata. So does everybody know what metadata is? Yeah, I'm getting nods. So information about data, so you can describe it. So that means things like ontologies, knowledge representations. I was working in the web, I did a lot of foundational work in the World Wide Web, because I remember the time before the web. Who remembers the time before the web? Two of us, yay. There was a time before the web. And actually you put knowledge into the web as opposed to just links into the web. That was called the semantic web, and now it's all knowledge graphs and large language models. I also did work starting in the 2000s around things like called computational workflows, which I'll mention shortly, which are ways of being able to stitch together steps in an analysis, so that all the heavy lifting is done by a workflow management system rather than you having to do it yourself using Python scripting. And I also co-founded the Software Sustainability Institute, big institute in the UK. And I was interested in collaboration. So I was interested in how do we change the way we do scholarship so that we share different kinds of knowledge. So we don't just share data or papers, we share data, we share methods, we share all sorts of different kinds of information. And how do we improve that sharing rather than trying to keep it to ourselves? So here's a little thing here about semantic web. So and I dug out this picture from a slide deck I gave in 2003, actually. So this is a long time ago that we were doing this. And so the idea is that you annotated the web, so you did annotations, so you described it using controlled vocabularies and knowledge in order to be able to link that knowledge across the web, so you'll be able to ask questions about the web and get answers as opposed to just doing point and clicking. Now, I mean, we have, this is what a Google thing I did yesterday is built in. People say, what happened to it? Well, it's built in. Every time you type into Google, you'll get a knowledge graph answer. That's because up to, 60 to 70% of the web is already annotated inside it with knowledge. Not just the words that's on the page, but knowledge about what the page is about is built into it. And that's called the semantic web. Does anybody recognize this person who's drinking with me? Do you recognize him, Christoph? No? Sam? That's Sir Tim Berners-Lee. He invented the web. So, so that's the guy who invented the web. It had to be invented by somebody, and that's who it is. And he's one of my friends, obviously. So, so this is like, again, another kind of touching the invention of things. So, and he actually, I gave I'll come across to that a little bit later on, but anyway, come back to that. So how did I get into bioinformatics? Well, it's people again. So this is a guy, Andy Brass, Professor Andy Brass, and he's a professor of bioinformatics. And in 1995, so we're talking 30 years ago, when bioinformatics had only just started. Actually, it was really the very beginning. He's one of the first people to have taught bioinformatics as a discipline. He said, I've got lots of data to link together, and I hear that you do this knowledge stuff because you're working on the web and things. Can you help? And so we did a project called Tambis, which was a project funded by, in the first wave, to try to say, how could we use traditional kind of clever database techniques and clever knowledge techniques in order to be able to have an automated system that you just posed a question and it would figure out by going to the different databases and linking them what the answer would be. Yeah, like, whoa. But it was early days, right? There weren't the huge number of databases that we have now. And it was incredibly influential. It was actually sponsored by Zeneca before they became AstraZeneca. Right? So it was incredibly influential. Of course, it founded some of the early ontologies of linking, of bioinformatics, actually. And but it took us 2 years to understand each other, because I'm a computing person and he's a biologist, and we're quite different. And the guy, Robert Stevens there, who I hired in as my first postdoc to do this project, he was the interlingua. He had a first degree in biology, but then he had a master's in bioinformatics and a PhD in computing. Right, so, and the reason he did a PhD in computing is because when he was doing his PhD in biology, he went blind. So he couldn't be a biologist at the bench anymore. So he became a bioinformatician early on. Right? And then 21 years later, he was my boss because he was the head of the biggest computer science department in the country, in the UK. And he was still blind, obviously. So, and he's one of the best biontologists in the world. Now, this is one of the reasons why all those if you do ontologies, one or two of you might do that. There's some talks later on in the conference about that. They have a back door because they're usually done visually. You usually describe them visually, and I'll show you that. So, like, they normally look like this. You draw them visually, but they all these tools have a back door because you can't see them. So it's done through language instead. Anyway, so this is what we were doing. What we were doing was trying to link and harmonize across all of these different silos of databases and have different ways of doing standardized reporting. Because I'm sure many of you have said, okay, here's a table with a header. What does the header mean? I don't know. It says, column 1, column 2, column 3. That's no good to anybody. And I don't know what the values are in the table either. So we had to have ways of standardizing the reporting and standardizing the language that we used for the reporting. So these they're like control vocabularies. This is the gene ontology. And the gene ontology was really established as a result of I think it was the 1996, something like that, ISMB, because they realized they needed to have a way of describing gene function, and so they started it up. So it's these kind of control vocabularies mean if I've said this term in this database and you've said the same term, we can link them together and we can build knowledge. So because this is what we're trying to do, isn't it? We're trying to link together things. And how do you link together things? Has it got the same identifier? So is it the same thing? And has it got the same description? Has it got the same tag? So we do it, a lot of times we do this sort of standardized reporting and so on. And so what we did was use the languages that we developed for the web and applied them to biology. That's what we tried to do. And that's what we did. Different ways of describing and annotating them. So we did that first kind of amazingly sort of in some ways naive, but on the other way, we didn't know what we were doing, so we could do brave things, trying to do a sort of universal integration system for all biology. But then we realized, well, that's very difficult. So we were much more kind of to scale. You had to be much more pragmatic. That's the difference between computer science theory and production. Being translational is being pragmatic, taking the ideas and then simplifying them so they work at scale. So that's the kind of thing that I do. So we did things like hide how you do this kind of tagging using these ontology terms in spreadsheets. So you don't know you're doing it, but just by using the spreadsheet, you're doing it. So we did kind of things like that. So that we can make things processable. If you do controlled vocabularies, if you do controlled tags for things, if you give things identifiers, you can make it processable by machines. And that's what we're interested in. And the semantic web was, how do I make the web understandable by agents, by AI agents, but by machines? But it was like 25 years or so, actually 30 years before we actually managed to achieve it. And we did all sorts of other stuff, including the early knowledge graphs. So that was cool. But then we realized, to really integrate things, what people do is pipelines. What they do is build these things like on the right. I've got some data and then do some analysis. I pass the data to the next step. I pass the data to the next step. And how do I do that data flow? And how do I control which steps to do? That's what people really do. Right? That's scripting that people really do. And later on, you're going to do some scripting, I think. Now, back in the day, we're talking about a long time ago. From 30 years, we've now gone to 20 years. What people did was do that by hand. And they used to do it by having Post-its. You see Hannah at the top? There were some Post-its. That's what they used to do. To say, I've gone to this database. I've got this. Now I'm going to cut and paste the results and I'm going to paste them into this one. And then I'm going to do the next bit of analysis. That's what people really did, was do cut and pasting and little bits of scripting. And, you know, does this scale, people? No, it doesn't scale. And that's because the databases were kind of designed for you to look at them with your eyes, see an answer, and then they didn't think about things like access through APIs or things like this. So this is what people actually did. They remembered what they were doing, or they remembered what identifiers were by putting them on Post-its and cutting and pasting to interlink. Well, that's no way to, you know, carry on. What you need is this automated computation stuff, right? And that was the next thing that we did. And we worked with all these people here, things like identifying biological pathways in resistance to in cattle. This was a sort of tsetse fly infection. And we built systems. We built a system called MyGrid that did this. So you see this all the time now. These are all examples of these kind of chain step things that people do in bioinformatics, for metagenomics or ligand simulation or high throughput sequencing or genome annotation. This is what everybody does, right? So how do we make that better? So we built a system called Taverna, and we're talking about 2002. We built this system to do those different steps, to automate that. Then there were about 5 systems that, you know, kind of did that sort of thing. Now there are 350 different systems to do this. But the big ones are things like Galaxy, Nextflow, SnakeMake. They're the ones that biology uses. And you'll have seen that there today, there were tutorials in those, and you'll see other talks about those. But there's at least 350 odd systems. And there's even a common workflow language to describe, to try to describe them all. And at the same time, we realized we really had to improve how people did software that was actually reusable, and software that was actually where you could reproduce what you did, and it recorded it, and actually had some quality associated with it, and still worked tomorrow, and didn't require you to download the developer as well as the software in order to make it work, right? So that's where we established things like the research software engineering community, which is a big community now in the UK and now internationally. And at the European level, we've established the European Virtual Institute for Research Software Quality. So that's where that came from, driven by the biology community in our work there. A combination of computing and biology and bioinformatics. Then we had the next request I got. We have a lot of systems biology collaborations. This was in 2008. How can we get them to collaborate, and how can we get them to share their data and models? So in systems biology, you have your data, but you also have models, ordinary differential equations, partial differential equations, now much more AI type models, but they're mathematical models that try to simulate metabolic pathways. And so they're software, really. And you have also standard operating procedures about how did you get hold of, you know, what was the experiment you did to generate the data that you're then including in the models. And these are all combinations. And modelers and biologists are not the same. Modelers are mathematicians, typically, very driven by mathematics. Biologists are bench biologists. They don't have the same ethos for sharing, right? This is a picture from back in 2009. You know, Mike Ashburn, a very famous bioinformatician, said biologists would rather share their toothbrush than their data. And he was right about that. They would rather share their toothbrush than their gene name, actually. Whereas modelers came from the computing kind of world, typically. And the computing world has much more notion of sharing open source. So what would we do there? So what we did was we built hubs. So we built hubs for sharing these workflows that I told you about, because they're kind of recipes for how do you do things, right, with software engines to execute those recipes. So we can share them and we can run them. So it's called, the latest one's called Workflow Hub that I run, which we started in COVID. It's part of that COVID thing I told you about. We also built an entire platform for doing all sorts of sharing in health, in industrial biotechnology, in bioinformatics, systems biology. So we built a platform for being able to share models and share data and to share processes and to describe them using these ontologies, to describe them using these semantic approaches, knowledge technologies that we developed for the web community originally. And so all of this is all stuff that we've built with teams, not just my team, but across globally as global teams, because this is joint infrastructure developed jointly across different countries in order to be able to build our foundational work. And then finally, I think this is my final bit, this bit. I work with a guy from the Netherlands, Baron Mons. He said, how do we stop burying all our knowledge in text? So when we do all this cool stuff, right? We do all this data, we do all this linking of data, we do all this sharing of, describing and linking of models and sharing of models, and then we write a paper about it and we throw it all away, or we bury it in a paper somewhere, or we bury it in different repositories, and then when you come to read it, you have to go and find it and bring it all back together. It's this like this we're back to the iceberg. Here's the article at the top, and underneath it is all the software and the methods and the data. So instead, how do we take the data and the tools and the code and everything, results, and combine it so that it becomes a unit that we can share when we share knowledge? And that is metadata plumbing. That's being able to combine the information, the descriptions, and the linkage between different things, like the different kinds of data, between the different repositories and different ways of doing analysis, and combining it. You see here publications, data, metadata, logs, and so on, packaging it together like in a factory, in a pack, so that when you get sent the pack, you can unpack it and go, aha, that's where everything is, and I can go and access it. And that's called research objects. And there's an approach called research object, or RO crate, that does this. We've been doing this now. This came up in, we started doing this about 2010. And now, 15 years later, it's really taken off, right? Because this is now a technique that's used widely. Galaxy implements it, SnakeMake implements it, Nextflow implements it, Dataverse implements it. All these different, the latest ZAR way of being able to interchange bioimaging data, that implements it. It's a kind of standard way of being able to package things together to implement in repositories and for systems. But it's based on that semantic knowledge technology that we did. And I've highlighted a couple of people here who've been involved. And all of this has been driven by this notion of something called FAIR. So FAIR means findable, accessible, interoperable, reusable. So the FAIR principles were kind of presented in 2016. Baron Mons, that guy I mentioned, led that kind of activity. He came up with the word. It's nothing new. It's nothing that you can tell we were doing this in 1990 blah. We were trying to do FAIR. We just didn't have a good word for it. But luckily a Dutchman came up with a really good word for it. And those FAIR guiding principles are now for software, for data, for workflows, and so on. And I'm one of the authors of this, and I'm the authors of the workflow people. So how do we bring this FAIR stuff to the rest of the world? Well, we have knowledge, research data management toolkits. And we have research software toolkits, which basically are how do we gather knowledge from across globally. People come together to do a mass collection of the knowledge of how to make your data better, how to use all these different tools, how to make your software better, how to use all those different tools. And Elixir, if you remember Elixir, that big organization, is really driving this activity. We have the Research Data Management Toolkit has over 200 contributors, people who have contributed to writing the pages about how to do this for bioimaging, how to do this for marine metagenomics. This is people wrangling. I talked about software engineering, and now I'm talking about people wrangling. This is how we wrangle, and I'm wrangling these two with colleagues, right? I always do all my work with colleagues. I don't think I've published a single authored paper, because everything is joint, right? And how do we make everything fair? And also, how do we make, how do we teach people? How do we skill people? So we've also established, for example, in the UK, a stewardship network. So people who do data stewardship, who do software, how do they get together and create a club, in order to be able to propagate these techniques? So here we are. That's how I did it. Or that's how we did it. We did knowledge and metadata, we did software and services, and we did collaboration. That's what I do as a kind of just my job, as it were, but driven by biology with biologists. And because that's cross-cutting, I also work in biodiversity, heliophysics, digital libraries, health data, climate change, particle physics, social science. Because everybody says they're special and different, but actually everybody is combining data, describing data, linking data to do analysis, trying to get people to work together, to share their data, to share their models. And increasingly, biology is having to combine with environmental data, combine with social data, in order to be able to really answer the big questions of today. So I'm not so sure about biology combining with particle physics, but in other areas for certain. So were there any lessons from all of this that I could impart to you? Well, this is what I actually ended up doing. I was doing theory in the 1990s, and in the 2000s, I was working with how do you put that into practice? How do you really do that for biology and bioinformaticians? Making things simpler in some ways from the technology, but making things harder because people are involved, right? So it's never, many issues are not to do with technology, they're to do with people. Right? They're to do with getting people to include some simple tagging in their database, or to mark up their analysis with some words that means you'll be able to understand it, to document it. People are terrible at that. I'm hoping that AI will help us, because AI is much better about that. And then putting it into practice, really building things that people could use. All those systems I showed you have software engineers and data scientists and data engineers working with them so that they really work today, and they really work tomorrow, and they'll really work next week, and they'll be able to manage things like AI attacks from AI bots and things like this, which is, if you're doing computer science research, you don't worry about that. But if you're delivering for bioinformatics, you do worry about that. So here's a few lessons. Get mentors. So and collaborators, there's just a few. I've got lots of collaborators, lots and lots of collaborators. We've worked with all these people who've stimulated and given us hope. And these people on the left, these were my career mentors. They either got me started when I was very junior, because remember, I was only a teaching assistant, a teaching lecturer. I was never expecting to do research. They helped me in my career. So that one's Wendy Hall. She's the UK's United Nations representative at AI, but in 1987, she was a senior lecturer working in the web, and she helped me. And the people at the bottom, these people, Tony Hay and Doug Kell, they had vision as funders to get funding for digital infrastructure and to fund biology and informatics at key points. So it's okay having all these ideas, but if you can't get any money, it's very hard. And those people created the environment to get the money. So they were very cool. And here's some examples. You could probably spot some of them in my talk. Say yes. So lots of people came to me and said, you've got, can we work together? And I said, yes, let's do that, because I'll learn, you'll learn, and we'll do something together, and we'll do things together much better, because I'll turn theoretical ideas into practice. And we'll learn from that what needs to be done in computing. And lots of my colleagues didn't say yes. Lots of them said, oh, only if I can, yeah, that's a great idea, and now go away, because now I've learned what your problem is. So I'll go and do the theory over here. That's not a way to make friends. Right? So I said yes a lot. And I made friends, and I kept them. Here's the thing is, you don't need to know everything. So if you're thinking, I don't know loads of stuff about biology, you don't need to know everything. You need to know how to combine things. You need to know how to build teams. You need to know what you don't know and get allies, get colleagues, get partners, get collaborators, learn from them. Chance favors the prepared mind. So every time we saw that people were struggling about how to combine data sets and doing it manually, you could spot, you could do this automatically. And we know some ideas how to do that, so let's do it, right? And that's being prepared and spotting the wave, making the wave, and then riding the wave, seeing new opportunities from other disciplines. And you have to remember as well that computer scientists and life scientists are completely different animals. They really, we spent a lot of time learning from each other how to speak to each other. The other thing I would realize as I was preparing this is you have to be incredibly patient. Right? So here's a paper on the left, which is from this year, 2025, and it talks about challenges in sharing metadata, so information in omics, and formatting metadata in omics. And it says it's really hard to collaborate, and for collaboration, we need to do a much better job at describing things using standards, and we need to have a much better job at how to get people to really collaborate and work together, and how should all that work, right? 2025, right? And here's a paper from 2008, right? It says exactly the same thing, which I wrote. And here's a talk from 2007 I gave at BOSC, The 7 Deadly Sins of Bioinformatics. And when I looked at this last night, I realized some of the technologies have changed, but it's exactly the same. Nothing's much changed, because it's all to do with people. Actually, it's all to do with people and social and cultural issues. It's just different. AI might change that. That's an interesting thought. What will change because of AI? And I see AI doing a shift, making a big shift here. But that's another talk. So my final point is, if you're thinking about doing bioinformatics, or you've done bioinformatics, or you're reflecting on bioinformatics, there's no one standard route. You don't have to don't just think, oh, well, I need to first of all get my degree in biology, and then I need to go and, get my degree in bioinformatics. And there are other ways, there isn't a standard route. You can do things differently. You just have to be open You just have to be open. Your eyes have to be open, and you have to be prepared to listen to what other people are doing and think of it as a team sport. Okay, that's the end of my talk. I hope that was interesting. Was that interesting, Christoph?
There's a question online.
Okay, yeah, go on. Maybe you could just shout.
I'll just shout. So, an online user is asking for a closer explanation of the research iceberg, what exactly that entails.
Oh, the research iceberg. This one here.
Yes.
That one. Okay. So it's actually from I put a link in the slides to the paper because I'm a good academic, and all good academics put all their citations in. So these slides will be available, and so everything has the citations in it, so you can go and have a look at it. So what it basically says is, if I'm publishing a research article, I'm putting a paper out, right? There you are, it's a PDF, woohoo! And it has, maybe it's a preprint, maybe it's in a learned journal, maybe there's a presentation associated with it. But it's just a narrative record written in language, right? All the stuff that it's built on, that's what the data sets you developed, the reference data sets you analyzed against, the software that you used. What mistakes did you make? Did you report the mistakes? Of course you didn't report the mistakes. Of course you didn't report the proper standard operating procedure or the methods. You just said, I did it in the usual way, whatever that is, right? Because that's what people say. You're like, oh, there's only 200 words to write the entire method. Actually, the method is what really matters. Nothing about what was the research question you were trying to do, or did you actually shift the research question halfway through, and all that kind of stuff. Where can I get all this? Where can I get this iceberg bit so I can go and check? If I want to check, that's reproducibility. Or I just want to reuse, because you've got a cool new, you used a pipeline to do the analysis. Can I use that pipeline to do the same analysis on my data? And that's what this does. Workflow Hub has 1,200 workflows in it, right? And you can go to those and say, that's what I do. I'll use that one. Okay, and if it's Galaxy one, you can go, that's what I do. I can click on it and do it, because you can launch them from the hub. Or I can take that one and revise it, edit it, because most of those pipelines, right, they're just variants. They're just variants of what people do the same thing but slightly differently. So take it and rework it and re-register it so somebody else will be able to use it. That's what I mean by the iceberg underneath. And we need to expose that iceberg underneath in order to be able to be sure that the top bit's good, but also that's where the knowledge is, right? For us to all build on top of another iceberg, right? Does that make sense? Did that make sense to me? Oh yeah, it makes sense to you? Did it make sense, Sam, to the person? Did it make sense?
They haven't answered yet. Oh, there we go.
Did it make sense to whoever is in the world out there who asked that question? Who knows? Did it make sense to you guys? Yay. Made sense to these guys. Okay. Any questions here?
Wonderful presentation, thank you. I want to ask, because the majority of the presentation that you shared are regarding the various tools and I would say consortia that you've developed to further data accessibility. And so from the viewpoint of students heading into either computational biology or CS, right? It's quite, I would say, a steep curve for someone to go into these software and then work them out and then see 100% what you're supposed to do. And so as you've created several of these tools, what are your recommendations for us to kind of try out different things on these.
Oh, okay, that's a very good question. So all of these tools and everything that I talked about is open, because that's one of our mantras, right? Open and fair. So you can just go and download them and use them, but that's not the same as being able to understand how to use them, right? And that's part of the reason why I promoted this kind of, I don't know if I spotted it here, training, right? And because even bioinformaticians don't know how to use some of them, and biologists certainly don't know how to use some of them, and neither should they have to, actually. I don't think it's necessary or indeed desirable that everybody has to learn everything. That's not what, it's team-based, right? You have specialisms. So that's a very interesting question. So I think actually we really need to do different kinds of training, different kinds of support than we would do for people who were in active research, because these are just school students, you mean, or just student or university students?
I would say like a broad spectrum. You have people who, who do research projects like the student challenges.
Right.
And you also have students who came out of biology in high school who wants to work on something.
Okay, well then that's, you're right, it's not so straightforward, right? Because there's a lot of assumed knowledge, I think. So, and a lot of the, within Elixir, for example, I say we really address the postgraduate or the undergraduate level. So undergraduate, postgraduate curricula, not the school level. And because you really do need to have, I think, some support in order to get on board. To say, just go and use it, I think that's really naive, right? Because it's actually quite sophisticated, or maybe we don't think it's sophisticated, but there's a lot of tacit knowledge that's built in. So I think that's a very interesting point that I'll take back to my organization to say, what are we doing for schools? Because I don't think we're doing anything really for schools. For universities or, sort of a higher education, for sure. All of these networks, all those communities that I showed you about from Elixir, anyone can join. And I think it's really useful to think, I don't have to know everything when I join. I'm here to help learn. I learned everything through an apprenticeship. Okay, so apprenticeship is really, what we really do is find mentors. These are the people. I was apprenticed to these people. I think of them as my, think of it as an apprenticeship, sitting with people and learning with them. And then I'll come up with some idea and then we'll help. Things like Google Summer of Code are an example that we use. We have things called the Biohackathon, which hackathons. We do national hackathons. We do international hackathons where people can come together and program. And I think it would be very interesting actually to think about how would we do hackathons with just focus on students rather than just thinking about them in general. But yeah, I think that's a very thought-provoking question. Thank you for posing that.
Thank you.
Yeah, I need to get my arse into gear. Any other questions? Yeah, well, there's a hackathon now.
Thank you. Very, very inspiring talk, really. Really, I am wondering, because you talk about challenges about sharing data, which is a cornerstone for the data-driven science we're trying to do. You talk about the mindset we have. Yes, of course, we have to be open, making friends and so on. But then one of the challenges is that people is really not open to these things, and things get political.
Yeah.
And then I would say, what would be, I don't know, the suggestions, or what is your thoughts about making these things political? I mean, how do you become a fair person that convince others to collaborate? How do you convince others to collaborate with you, really? Because this is a very hard thing to do.
It is, and it's kind of hidden in this word here. And actually, there's a whole. I took out all those other slides because I've got whole talks on that. How do you, what's the word? How do you build this really collaborative environment? In fact, I wrote a paper in the late 2000s about how to do software collaboration with scientists. And it's really about reciprocity. So what I mean by that is, if I'm doing something and you're doing something together, everybody has to benefit. There has to be, we call it the jam model. Jam today, jam tomorrow, jam for everybody, right? So everybody will get benefit. It'll be incremental, and you'll have to do compromise, right? So I'll have to do some work which one of my colleagues might say, oh yeah, but that's kind of engineering work, that's not research. It doesn't matter, because I'm working with you. It helps you because you need to use it, but then you'll say, now we can scale this up and we can get impact. And I care more about impact than I care about academic papers. Like, I want knowledge in all databases everywhere. I don't want to just describe one in a very clever way. Do you see what I mean? So it's kind of understanding that reciprocity relationship. There's an entire research discipline called team science. I don't know if you're aware of it. And there's the International Conference of the Science of Team Science, which is in its 18th year. So there's an entire world of how do you get people to work together. But it's all to do with incentives and rewards and respect and being able to, everybody to benefit. Now, the key point here is that that isn't how academia is set up at the moment. And it isn't how publishing and scholarship is set up at the moment. The rewards is all screwed, right? So actually what I do now a lot of policy work at G7 level. I'm on the G7 Open Science Working Group, OECD, UNESCO, and in the UK, NIH mandates and things, to create the environment that means to change how we do research, to create that. Now, you thought this was a long game. That's a long game. That's a long game, because we're operating at the moment a 19th century reward and publishing environment in the 21st century. And so that's, again, people. It's people and culture. So, but we can have a long discussion about that. If you want, and I took it out of these slides, unfortunately, I would recommend you look at Brian Nosek's The Strategies for Cultural Change. He has a model, which I wish I'd left into the slide deck now, which says, in order to be able to really do what you want to be able to do, we have to have the infrastructure to be able to do sharing. We need policies that drive the sharing. We need skills and make it easy so everybody understands what to do. We need incentives in order to make it rewarding for everybody, all the stakeholders, and we need communities to make it normative. So you need all of those 5 things, and you need them all to be happening simultaneously. You can't just have one and then, because they will block each other. So you have to do it all simultaneously. And this is my North Star in all the work I do. But when I'm building one of these, when I'm doing infrastructure, or I'm running a committee or whatever, I'm thinking those 5 things, how are we getting them all to work together? Because you can't just ignore one of them and think it will work, because that will be the thing that fails it. Do you see what I mean? So Brian Nosek, Cultural Change, find that. It's good stuff. Okay.